I'd like to introduce Dr. Heather Lau. She is a pediatric neurologist and now Executive Director of Global Clinical Development at Ultragenyx. And so happy to have you here, Dr. Lau, talking to us today about the accelerated approval process.
Thank you. Just want to check that you can hear me. Great. So thank you for inviting me to speak today. It's always wonderful to connect with the community. As Kara said, I am now Executive Director of Global Clinical Development at Ultragenyx. But in my prior life, in my academic life, I ran the lysosomal program at NYU for over 10 years with a focus on MPS. So it's a great pleasure to be representing at Ultragenyx our program in MPS 3A. As many of you know, we acquired that program over 2 years ago from Abeona, and we have been working really hard to bring that forward. And so today's discussion is to kind of reflect and educate on this accelerated approval pathway. And I thought this would be a useful discussion since we have been talking about this pathway from Ultragenyx as we apply it to our own program for MPS 3A, but it's also relevant for all the neuronopathic MPS disorders. So I thought it would be useful to go through a little bit about what it is, where did it come from, and why we're leveraging this pathway. So let's go to the next slide. And just as a reminder, I'm sure most of you know that Ultragenyx was founded by Dr. Emil Kakkis back in 2010. Dr. Kakkis had already established a wonderful career in the drug development for MPS disorders. And then he founded his own company that's specifically dedicated to rare and ultra rare diseases. If you look at our pipeline, you'll see that we're not tied to just one type of treatment. You'll see enzyme replacement, you'll see small molecules, monoclonal antibodies, and now gene therapy. In fact, we opened our own gene therapy manufacturing plant over a year ago. So we're now grew, and I knew Dr. Kakkis back when he founded that company in 2010, and I've seen it grow into now a mid-sized company of over 1,200 employees, and we are global. We have a flagship location in Novato, California. I'm located on the East Coast, and we have places all over the world. And so we're dedicated to developing these therapies. And what we'll talk about is how we do this. And one of the pathways of doing this is connecting with the patient community. So let's go to the next slide. So our goals for today, it's really to have a discussion about what are endpoints and surrogate endpoints and biomarkers, how are they used or could be leveraged for an accelerated approval pathway, and how we can hopefully impact access to new treatments. And of course, just discuss how this impacts specifically our San Felipe OA community, since we are partnering with the community to help us move this program forward. Okay, next slide. Great. So we're going to start off a little bit about some definitions. So let's go to the next slide. So what are endpoints? So endpoints could be a variety of ways to measure some kind of outcome. And the US FDA, we're going to focus on the US FDA regulatory pathway today. They focus on validated endpoints, and that's a key word there, validated, to understand how a drug is working in a particular population. And those endpoints could look at both benefit as well as risk. And it helps us track it over time when we're investigating a new therapy in a clinical trial setting. And it is from that beginning clinical work that we need to understand whether or not our therapy is working or not, and what are the safety aspects. And so typically these types of endpoints are directly measured outcomes, and they really are to determine whether there's clinically meaningful benefit for their patients. And these endpoints can look at a variety of factors. In certain disease states, survival is an endpoint. In our rapidly progressive disorders, functional capacity, whether it's looking at brain function or other types of function, or the modulation of the symptoms or amelioration of symptoms. We'll also look at the risk if there's any development of new or worsening symptoms, as well as the safety of this in a clinical trial. So these endpoints are listed out as a direct measured outcome, and they have to be negotiated with the regulatory agencies. And we oftentimes incorporate expert knowledge or expert advice from our scientific community, our clinical community, and at Ultragenyx from our patient community to understand if these endpoints are meaningful to our patients and families. Now, the problem with these types of endpoints is that it's typically difficult in trials for ultra rare diseases. And I'll have a little bit more focus on neuro or neurologic endpoints since I'm a neurologist and we're working on MPS IIIA. Now, when we're looking at ultra rare diseases, it's really hard to understand if there's to develop a validated endpoint in an ultra rare disease. There's small numbers of patients. There could be variability in the disease presentation. There could be variability in the progression, right? So it could take a while to see whether or not we're impacting the disease course in a positive way. And there could also be multiple organ impact, especially with some of our metabolic diseases that not only affect one brain or affect heart and affect other aspects of the child's functioning. So let's go to the next slide. So that's the concept of a validated endpoint that's directly observed, directly measured. It's really tangible to a function or an outcome. Now, surrogate endpoints are, say, a step removed from that, right? So, and how are we applying these in clinical trials? So it's really difficult to validate an endpoint in a rare disease. And sometimes we're looking for other ways to measure therapeutic response to an intervention, whether it's we want to know sooner that it's working or that it's not working even. And so that we want to shorten that time to see a treatment effect, because it's not feasible to conduct a clinical trial over many, many years. Sometimes, especially in developmental disorders or in other disorders that take a long time to unfold, a decade or longer, it's really hard for a researcher to conduct that type of research. And we want to know for the community sooner rather than later if something's working, because oftentimes, especially in the neurologic space, we're looking at irreversible damage going forward. So we want to be able to understand before it's too late. So we might try to look for a surrogate or an intermediate measure or endpoint. So again, it's kind of temporarily, hopefully something sooner in that process to understand it, to get an expected benefit. So these may not be validated or they don't have, you'll see a little bit about the accelerated pathway, but at least expected. Is it connected to a clinical outcome so that we could tie it to a traditional endpoint potentially? And so what you'll see today is that, there is a path using these surrogate endpoints to capture that disease progression or improvement in a therapeutic trial to shorten that length of time in a clinical trial and hopefully the overall approval process when we go to the FDA. So what are biomarkers? Well, some of us are thinking of only fluid markers, right? Blood, urine, CSF, but there are non-fluid markers too, such as imaging. And it depends on the disease state, what is more relevant. So it's not tied to one specific type of biomarker. It could be blood pressure in a blood pressure trial for blood pressure medications. It could be the volume of organs, like in Gaucher disease, where they look at the shrinkage of very enlarged spleens and livers as an endpoint. Or it could be blood levels of blood markers. It could be blood counts, or in this case is CSF-HS, which we'll talk a little bit more in more detail. So cerebrospinal fluid bathes the brain, and it has some metabolites in there that we could measure potentially. And in this case, we're talking about heparan sulfate, or the glycosaminoglycan that accumulates in all neuronopathic MPS disorders. Okay, so those are surrogate endpoints. Now, how does that apply, and what is this accelerated approval pathway that you might have heard about? Let's go to the next slide. So this was established back in 1992, if you can believe it, by the FDA to allow researchers to use a surrogate endpoint, often a biomarker, to evaluate the safety and efficacy of therapies for serious conditions with unmet needs. So those are the other criteria, right? If this is a reversible disorder that's not serious, but it's a treatment, they may not allow the use of a surrogate marker. They want more direct directly observed biomarker endpoints. But in the case of a serious condition or even a rare condition that has no other therapy out there, we start to see this regulatory flexibility to use a surrogate endpoint. And so using that and showing to the agency or the regulatory bodies that this surrogate endpoint is reasonably likely to predict. It doesn't have to absolutely predict, but it should be connected to that clinical benefit. It would allow a company, a research group, to accelerate the path forward. And I'll show you exactly how. All right, let's go to the next slide. Okay, so again, this phrase is really important to us. A biomarker that's reasonably likely to predict clinical outcomes. It doesn't have to be absolutely validated. And it may be used, and this is exactly from the guidance, may be used for accelerated approval for drugs. Now, here's the caveat. So when you use a surrogate endpoint, you eventually have to show that it's connected to clinical benefit. So what does that mean? It means that they'll grant approval based on that surrogate endpoint, but then the sponsor of that trial needs to come back later. Now we're marketing and be able to get that drug to commercial access, but we are committed in some form to come back to the agency to show in either our evidence that there's confirmation of clinical benefit. And that all has to be negotiated with the FDA ahead of time. And so we can identify the surrogate endpoint, but then we also have to negotiate how we're going to show that connection, when are we going to show it, and how much information or how much data do we need to show. Sometimes it's in the form of another trial, and other times it's in the form of just long-term follow-up of patients. And this is all individually negotiated by each sponsor of each drug. So thinking about the ideal biomarkers, right? The ideal biomarker would be biologically plausible. What does that mean? It's connected somehow. We understand the mechanism, why that biomarker is deranged or abnormal in that disease state. So for heparan sulfate, why is that biologically plausible? Well, for MPS IIIA, B, C, and D specifically, that's the only glycosaminoglycan that gets elevated because those enzymes are specific to that heparan sulfate degradation pathway. And in fact, and for other neuronopathic MPS, HS is also implicated as neurotoxic. And so therefore, it would also be relevant there. So that's connected to the enzymatic defect in MPS IIIA and the others, leads to this accumulation of this substrate. And in fact, there's specific FDA guidance on that very scenario, substrate accumulation or deposition in the setting of a single enzyme defect. It's a lot of words, but basically you lack a protein to break down something else. And that could be used as a measure of therapeutic response, good or bad. And that's why we're putting that forward as biologically plausible. The ability to measure it needs to be sensitive and specific to that disease state for an ideal biomarker. Now, you'll hear about other biomarkers that are not intimately related to the primary metabolic pathway, but they're still helpful. But ideally, it would be very specific to your disease state. You should have at least a qualified way to measure it consistently and reliably. It should be relatively minimally invasive, right? We don't want to have to do biopsies. Now, lumbar punctures to get CSF might be invasive, but it's less invasive than actually directly measuring something in a tissue, such as eloquent brain tissue. It should be easy to measure, generalizable, and relatively inexpensive. Okay, so those are the ideal characteristics of a biomarker, which is really hard to meet that in a rare disease state. So let's go to the next slide. So again, just to summarize, why do we use this in rare disease? Well, we can show, per the guidance, it's extremely high unmet need in rare diseases. It's very often we don't have any disease-modifying therapy, right? We have symptomatic care or symptomatic treatments, but we're not really getting to the root cause. And there's an urgency in the rare disease community to allow research to progress at a faster pace. So the rarity of the disease, low numbers, lack of other clinical trial data, especially natural history data, it might be slowly progressive, or at least slowly as compared to a standard trial of 1 to 2 years to see how a therapy is potentially impacting the natural history of that disease. And there might be a delay. There might be a period of normal development or very little bit of disease burden and then the onset. Can we measure that biomarker and it's elevated or abnormal before there's actual onset of clinical symptoms so that you could prevent irreversible damage, which is really important in the brain, right? Before we have neuronal loss, can we measure something? And in fact, we can. We can measure heparan sulfate. It's elevated from birth. So that's why, our company and others and in the community you'll hear about got together to talk about the relevance of CSFHS, especially before there's irreversible damage in the brain. And then there's other lack of validated endpoints, as I described before. Okay, next slide. So the traditional path, which again is not, should probably extend this out very far, but it really starts with basic research in the lab. In the petri dish, in cell models. Then it might go to animal models, preclinical studies, and that jump from preclinical to clinical is a big jump. Before we need to understand something about our drug before we even attempt treatment in a human, right? We want to understand the safety to the best of our knowledge. Then those clinical trials, really, we can expand that purple box very far because those clinical trials in a traditional approval could take years, especially if we're looking at the brain and development, to really see a clinical endpoint that changes over time, especially if there's a window of no symptoms and you want to understand it and we treat early. And then from there, we have to have those regulatory discussions. We move forward and we go for FDA approval. And then eventually, once it's approved, you still don't have access because now we have to talk to payers. We have to understand how to get the drug out to all the different hospitals around the world, or in this case, in the US. And then there's a whole other concept of reimbursable. Can we get this for insurance companies to pay? And so on the market is also a big jump. To not only get the approval, but then approval with the ability to disseminate the drug, get it in the hands of people that can prescribe it, and get reimbursement for it so that it's not a burden to the families. But this process can take 15 years. So how do we accelerate it? Next slide. So it looks longer, but in fact, what we're showing here is that at the clinical trial where we are right now, we're in our program in the phase 3 portion. In the midst of negotiations, we sent a press release last June that the FDA did align on using CSFHS as a biomarker for accelerated approval. And so those negotiations help us to understand if we can go faster to market authorization. So we don't have to show a full readout of all the clinical endpoints before we get, quote, conditional approval. And that approval then would allow us to go to market, and meaning get to commercialization and market and get it out there. Because right now the drug is only available in the form of in studies, right? In research. So we want to get out to the commercial market so that we can get access across. You don't have to be a specific site, but you would have access to it. But here's the caveat. This acceleration could take years off of a full approval, but we are then negotiating the commitment, the post-marketing commitment. So you'll see 2 markets or 2 market authorizations. So we go back, and at that second green box, we need to go back with more data now. So patients are getting access to drug through the accelerated pathway. They don't necessarily have to be in a clinical study to get the drug, but there are negotiations that have to be had to say to the regulatory body, we're going to come back and show you that that biomarker really did translate to a clinical benefit, and here's that clinical benefit. Now, if you look across different programs and different drugs for rare disease, that can look very differently. So don't assume that because sponsor A had to do it this way, that sponsor B has to. There's some lack of uniformity. There might be different types of, it might be a different type of drug. So it's individually negotiated based on the not only the disease state, but the type of drug, and specifically, our negotiations. Now, we're trying to be as transparent as possible as a company so that others can learn from this process as well. But this part is where it's really critical that we do rely on the patients that are getting the treatment to understand over the longer term, not just safety, but the long-term benefit. And then we have to go back to them and present that data again. So yes, it's faster to market, but we are then committed to coming back with that full data, that full clinical data set to understand and prove to the agency. Because here's the caveat: they can pull the approval at that point if we don't show that that biomarker led to a clinical benefit. So it's a risk, but the risk is offset by the acceleration to market. Okay, so let's go to the next slide. I think that was really important. And that's why when you're thinking about a biomarker, we really want to understand if it's truly reasonably likely to predict a clinical benefit. And individual companies were working in the space to negotiate that CSF-HS is relevant. It's a relevant biomarker for a variety of neuronopathic MPS disorders and for a variety of types of treatments. So not just gene therapy, but for enzyme replacement and other therapies. And so this was a non-decision-making forum, meaning it was a third-party group called the Reagan-Udall Foundation that, Emil, Dr. Kakis, reached out to, as well as to other companies in the space. And then we extended it to our academic clinicians and researchers, and then most importantly, our patient community, to really come together to look at what we're all doing individually and try to come together to show that we all are aligned that this is a biomarker that's reasonably likely to predict clinical benefit. And so pre-competitively, cross programs, cross sponsors, we held a forum in February, and you've heard some of that today. And Kara was there, as well as others from our patient community and our researchers, our clinicians, Dr. Jones, to talk to the community. And over 100 FDA members were also present. And that helped us kind of get across to the FDA and others in the community that we're aligned, that this is a consensus. And that really helped to open the door. So even though there was not a formal decision from the FDA, it was a forum for listening and understanding and showing them that there was a consensus. And I felt that, that helped not just Ultragenyx, but others in the space to accelerate. Next slide. Okay. And so how does this happen next after accelerated approval? So I'll share with you a little bit of our personal journey to highlight what are the next steps. So we've gone through our phase 1 and 2, that was under Abeona. Phase 3 was evolved under Abeona. Ultragenyx took over in 2022, and we've been compiling that and looking at that data. We aligned with the Reagan-Udall Foundation. So this is starting in February, and partnering with the community and other stakeholders. So what's the next step now? So we got a positive signal that CSF-HS is a biomarker that they would look to as for accelerated approval. So now we're looking at and trying to understand all that data, and we're compiling that data and all of those analyses of all over those years with Abeona, and then our time managing the study. And we are now creating what we call a biologics license application, a BLA. And that process is comprehensive to show in the FDA all of that data that we have to date. So even though it's accelerated, it's still a lot of data to show safety and efficacy. And it goes across not just clinical, but manufacturing. And then this is the regulatory path. And so that takes time to bring that dossier together. And during that time, in parallel at Ultragenyx and other companies, people will start to look forward and say, okay, if we get market access, well, we have to be ready. We have to have discussions with payers, healthcare systems, communities to understand, are they ready? Are they prepared for a novel therapy in a rare disease? Are the payers understanding what the value is? What is the potential of this? And that's in parallel. And as we move forward, then the FDA, we submit that BLA, and I think it's public knowledge that we are attempting to submit within the year. And so the next step is the FDA reviews it themselves. And that's where we go silent a little bit, where the FDA will look at all that data. And we're continuing our work with planning ahead, whether it's for manufacturing, more material, looking to expand and understand how we can have a access across institutions. they're reviewing that. Now, there's two paths. There's a priority review and a traditional review. Priority review has a little bit faster review process, and we don't know either way. We will not know until later if it's a priority or traditional. And then if it's priority, it's a little bit accelerated. And then during this time that we're waiting, there is an opportunity or potential that the FDA looks at the dossier and asks, well, we need more advice from the public, and there could be a public advisory or AdCom meeting. You might have heard about that in other disease states, such as for Duchenne's. And that's a different process where the FDA is engaging with a community and asking for advice on an aspect of that. It may not be the whole approval, but a question might be asked. It's not a given that it always happens, but it's a potential, and we have to prepare for that as well. And we do see that we have patient advocacy, caregivers and patients, as well as other sponsors and others in the space attend those public and might testify at those AdComs. So that's not a given that it will happen, but we do prepare for that. And community members are really critical to these FDA advisory committee meetings if they're called for. And then we get to the approval. So then the FDA determines approval under the accelerated approval program. And once again, that goes to market access. And that's a positive result. But in the background, remember that confirmatory evidence needs to be collected. And that means we need to stay in touch with patients and families after they receive the therapy to understand if there's a longer term clinical benefit, over the longer term. And then we have to go back to convert to a full approval. So that's a really long process, but hopefully it's useful to you to understand that an approval doesn't translate to immediate approval, nor does it translate to a full approval. They could still pull a drug off the market if after they show the confirmatory evidence does not support clinical benefit, and that's a risk. Okay, next slide. So I kind of already said this, but approval doesn't always mean immediate access, right? So we talked about how in the US, we have to then understand how the regions within the US can get access to this. Are institutions ready for it? Are physicians ready for it? Are they understanding what it is? The payer system is really another layer here in the US. And those are not a unified, right? It's not a one payer system. There's small entities, large entities. There's different insurance companies out there that have to look at this and understand how they're going to deal with a new therapy in that space. And I'm not sure if you've gone through that, but as a clinician, I went through that all the time, getting authorization and approvals, which vary based on the insurance company. Now, other factors might play into the payer decision. It also might depend on what the label, right? I didn't even talk about the label. Is it, a focused label? Is it an expanded label? Who will have access to it? And that's also under negotiation. And so the good news is that can evolve, even though there might start off with a certain coverage initially, it could expand over time to a broader coverage. Again, these are very technical, but very impactful because it impacts you, the family, of whether or not you have access to that therapy right away. And these are so many different factors that we have to work in parallel as we're trying for this path to access. Okay, next slide. So I probably went a little long, but I thought this is important to communicate to the community that this next phase, there's the initial research that is really fascinating and preclinical, and it's giving us hope. Clinical studies give us now an understanding of safety and potential benefit, but then it's still a long road after. And so using accelerated approval based on biomarkers is really important for rare diseases. It's important to understand and identify a biomarker that's relevant. That could be elusive, especially in neurologic disease. It's really hard to find a brain relevant biomarker in other disease states. And so it was really partnering with the patient community, the researchers, the clinicians that really helped us in MPS move this forward for our neuronopathic MPS disorders. And really and truly, accelerated pathway is the first step. Confirmatory evidence is critical so that those therapies can remain on the market once we are granted accelerated approval, in order to stay on the market and stay having access, we need to show that longer term benefit. And that relies on confirmation of clinical efficacy in the patients who receive the therapy. And then once we have, once that treatment is approved, we will work with the community, and we're already starting to understand how we can get a broad access to this therapy. And we really, really do rely on Cure Sanfilippo and MPS Society and others to help us do that effectively. So thank you for the time. If you have questions, I can't take questions today live, but please reach out to our patient advocacy team. Jenny McHugh is here currently with us. Kristen Voorhees is on leave. You might know Kristen as well, but please email Jenny and we will try to, in a expedited way, answer your questions. Thank you so much for this time. Really appreciate it.